My name is Sohan Jeeta. I'm the founder and chairman of NoRCEL, Network of Researchers on the Chemical Emergence of Life, as Savio said. And I've got a few things to share with you guys. They're very important, to be quite honest. So the first bit is that this is where we are with our network. So these are the ones that are active currently. We've got Frontiers of Sciences, Sessions, as Savio said, and then Microbial World Network. We also got the Astroscience Exploration Network that's active in Africa, whereas the Microbial World Network, that's active, run by Vice President of NoRCEL, Oleg, and his colleague, Vladimir. And then we have the Latin American Hub, which is run by our friend Miriam, who's present in this forum. And then there is the latest one to join us is Southeast Asia Hub, and that's C. And it will be run by a PhD student called Mohindran from Malaysia. We are currently talking to Turkish people, and I'm holding a Zoom on Saturday, and they want to come on board as well. So they want to run their initiative. Meanwhile, West African, that will be a girl called Philippa from Nigeria. She will be running the West African initiative, and it'll probably be called West African Hub, WA, you know. And but she's running it by her boss, and I'm using her name, by the way, rather than saying supervisor. Sorry about that, but anyway, it just stuck in my mind. So this is where we are. We're kind of innovative people. These are the innovative initiatives from NoRCEL. I'll come to that point for why they're important in a minute. And of course, if I am forced to define what we mean by it, what does NoRCEL represent? You know, initially we started off with the idea that we're a network of researchers on the chemical emergence of life, which is still true, which is still holds. But I have actually cut it down to one word. And I think it is a movement, as shown by the previous slide. You know, as shown by this slide, you can see that different people are doing different things, and different people are doing different things. But by the way, I think there was oh yeah. And here's another thing to show you what we mean by its movement. And we have evolving education being launched in Africa now, specifically in Nigeria. We're beginning in Nigeria, but it will be unrolled across Africa in time. And Astroscience Exploration Network is the hub I'm talking about. And so what we're doing there is introducing science, space science curriculum. Currently, I'm writing that. I've written 3 so far. I've got about 7 more to write units. And then we have setting up, I missed the word. I only just done this PowerPoint just before you came on board, and that's why I was late. So setting up is missing. Setting up an astrochemistry lab in Nigeria as well. So if we're going to give them a science education curriculum, we have to give them hands-on experience as well. So this is kind of backup doing for hands-on experience. Then we are arranging also remote sensing telescopes where people can access these and carry out various observation in further afield beyond our solar system. And then, Oh, there's something missing actually. I can't seem to see my top of my bar. What's that? What's that? How can you help us, or how can you help? I'm talking about about all the young people here today. What they can do is to be part of the movement and promote the science the way you feel it should be. This is because you saw at the beginning, we got various networks. All the people are running individually. You know, like Savio is doing his thing individually. Miriam's doing the same thing. So anybody can do anything. And we're just forming a little groups where we are promulgating science. By the way, NoRCEL is mostly active in the global south. So also, you can actually help by becoming a member, which means that you can contact Savio. He's the main man on this this forum. You can contact Kathy at the administration, and you can contact me, and you can get the details from our website, which is norcel.net. And that's all I have to say. Thank you very much for this, Savio. And I'll just come and share now. Stop sharing.
Okay.
Yeah, thank you, everyone. Thanks.
Thank you. So, Martin, can you say something?
Yes, I could add something to that. So, hello, my name is Martin Dominik. I'm the president of NoRCEL. I'm a researcher at the University of St. Andrews. I'm also co-director of the St. Andrews Center for Exoplanet Science. Now, NoRCEL, the network of researchers on the chemical emergence of life, as the name promises, it is a network. And what makes any network is its members. As Soren already said, NoRCEL is about you. There's lots of you about NoRCEL, and others within NoRCEL are there to support you. What further characterizes NoRCEL is that we aim for transcending disciplinary confines. And we enjoy good discussion, making efficient use of our diverse expertise. We really want to hear views. We want to encourage collective endeavors to explore new directions. In order to get there, we embrace critical and creative original thinking and thereby inspire each other. I've learned so many things in all the years that I've been involved, and Soren just reminded me it has been 10 years.
Yep.
Every time I look at scientific journals, it's kind of striking that I find so many articles in which the phrase, "it is well known that," is followed by a statement that is plainly wrong. At NoRCEL, we do not hesitate to question, rather than tacitly accepting widespread belief. And as Soren has already indicated, and you might have already noticed, for us, international network does not mean just Western Europe and North America. We are actually there where the creative potential lies. And it's not within just a few institutions amongst the highest developed countries in the world. So, if you want to go beyond the boring stuff, you've come to the right place.
Hi, Mark. Thank you so much, Martin. Now I have the pleasure to introduce Dr. Alan Templeton. And Alan has worked in many fields of biology, including human evolution, population genetics, and conservation biology. And in all of those fields, his studies have had a profound impact. And ladies and gentlemen, and it's a pleasure to be introducing my friend, Dr. Alan Templeton. Alan, thank you so much for accepting my invitation. Please, now you can share your screen and please go ahead.
Okay. Well, thank you for the invitation. You know, I've been coming to Brazil, I realized, since 1976. So I'm kind of sorry this is online rather than in person, because eu gosto muito do Brasil. Okay, so let me start. Let's see. Is anybody seeing my slide?
Not at the
No.
moment.
No, your slide.
Not yet.
Not yet.
Okay, share screen. Okay, now I think it'll come on. Okay, does everybody see it now?
Nope.
No.
Nope.
Yeah.
Oh, yeah.
Okay, great.
There you go.
Okay,
If you-
so I'm going to talk
if you
about,
can- Sorry, could you
Yes.
flip between the presenter's view and the other one?
oh, which view are you getting?
We're getting the presenter's view.
Oh, okay, let's see.
Yeah,
Okay.
That's
great.
better.
That better?
Yes.
Okay.
Great.
Okay, no problem. Okay, no, I'm going to talk about a project in conservation biology, but it's also one that hits some politics as well. And Martin will like this because at the end I'll talk about things that are well accepted and generally known, all of which are false. So I think he'll like the last couple of slides. So what I'm talking about is at first a simple conservation story. When I'm talking about this Heinz emerald dragonfly, which is a very beautiful dragonfly. In fact, dragonflies are beautiful animals to begin with. And it's one of the few insects that has made the federal endangered species list in the United States. The Endangered Species Act is heavily biased towards vertebrates if you're an animal. And so even though there's many insects that probably should be on it, there's only a handful, but the Heinz is one that did get on it. And I'm going to talk in particular about a problem about what are core populations and peripheral populations and the impact of those in a conservation program. So first of all, I want to let you know who has been involved in this. And this is important because, in fact, of the people on this long list, there's only 4 that you would think of as scientists: Meredith Mahoney, myself, Everett Cachet, and former postdoc of mine, Stan Browdy. And all the rest of these people are basically in some other occupation, okay? In fact, the head of our group, the one who really organized this and got everything going, Jane Walker, who unfortunately died very recently, but she worked for Walmart, a very large retail chain in the United States. So this is also an example of citizen scientists, so to speak. And in fact, this work could not have been done without all these people who volunteered their time, even their money, certainly their vacation days, okay, to make this project work. Okay, so I want to emphasize the important role, particularly in this case, because getting funds for insects is very hard, even in the United States. So without all these volunteers doing all this hard work and donating much of their time, this project could not have occurred. So, okay, why is it not advancing? Okay, so first of all, what is the geographical range of this insect, and why is there a core peripheral kind of dilemma? Okay, so this dragonfly is specific to fens. So fens are wetlands, especially where water seeps out of calcareous soils. And anyway, they have their own unique attributes in terms not only of the insect life, but the plant life and other associated life forms. And most of the fens in North America are found in what we call the Great Lakes region. So the Great Lakes are just surrounded by many, many fens. And each of these dots represents our major sample sites. Each area often has several fens within it. So these are not the number of fens they're found on. And in fact, you can see the bulk of the range still today is in the Great Lakes region, going from Ontario and Canada into the United States. We know there were extinct populations. There was a lot of collecting of this species in the 1920s. So we have museum specimens, and many of them came from northern Ohio and Indiana. But those populations are all extinct. Basically, a fen is a very easy habitat to destroy. The farmer can ditch it and destroy a whole fen in an afternoon. And so yes. So anyway, a lot of these fens in this area here have been converted to farmland. So there just aren't any fens now anymore in this area. There was one specimen, a very isolated one, found at the very end of the Appalachian Mountain Range, which goes down here. It was found also in the 1920s, but it's never been seen there again. But there is another peripheral population, so-called peripheral population, in the Ozarks. So right now I'm speaking to you from right here in St. Louis. And so if you go kind of south and west from where I am right now, you'll hit a highland region called the Ozarks. And so even here, it is much more geographically restricted than almost any of these dots that you see here of the current populations in the Great Lakes region. So in the Ozarks, here we have basically, these are all the sites, and we've really looked extensively at the Ozarks with all these volunteers. And so we're pretty confident we have most of the sites this species still lives in. And once again, each of these sites can actually have multiple fens in it. So these are not the number of fens, but rather the number of general sites we have. And most of them are along the tributaries or this river called the Black River. It's a major river coming out of the Ozark Highlands. And it's got lots of endangered species in it, also the current river, including these dragonflies. And so we have these, but you can see the bulk of the range is right here along this Black River, Current River axis. And there's one population very far apart over here. But the bulk of the range just in the Ozarks is limited to about 100-kilometer transect. So this is a tiny, tiny area compared to the Great Lakes region. So I mean by multiple orders of magnitude. And moreover, the fens in this region are very small. As I'll show you later, they're about 2 orders of magnitude smaller than the fens you have in the Great Lakes region. So before I go on, let me talk a little bit about fens. So here is a picture of one of my granddaughters crossing a fen in the Great Lakes region, actually in Ontario. And you can see it looks kind of like a grassland, but it's really not. She's on a boardwalk for a reason, because if she stepped off the boardwalk, she would sink up to her waist in mud. You can still see a few areas of open water, but even the areas that look like they're covered with grass are really, there's water there, often standing water, but hidden by the grass, and there's a thick muddy layer. And that's very typical of the fens we find in the Great Lakes. And it's also the same true for the Ozarks. So here's a fen in the Ozarks. It looks very much like a grassland until you walk into it. And then you quickly find that, in fact, it's very muddy and a lot of water is in it. And so there's several species that live only in these fens. And in fact, this dragonfly is in a mutualism with a particular kind of crayfish. And so you need both the crayfish and the dragonfly in the fen for both of them, or at least for the dragonfly to survive. Now, because it was federally listed as an endangered species, the U.S. Fish and Wildlife Service became put in charge of the protection and survival of this dragonfly. And in 2013, after some initial studies that had gone on for almost a decade, mostly in the Great Lakes region, the Fish and Wildlife Service recommended that 2 recovery areas should be established. And those were the southern and the northern Great Lakes. And the Ozarks were left completely out of the recovery plan. There was not a single effort to be put to protect them in the Ozarks. And this upset plenty of us who were on the Ozark subcommittee, so to speak, of this species, including myself. And so we had discussions with the Fish and Wildlife Service people, and it revealed that they had a very strong rationale for excluding the Ozarks. They said it's just a peripheral population. It's found in a very tiny geographical area compared to the bulk of the species range. The fens are tiny and teeny tiny there. And that means the population sizes are undoubtedly small. There's no genetic diversity in the Ozarks. We have limited resources. We're not going to waste them on a peripheral population. We're going to concentrate on the core. So that was the rationale of the Fish and Wildlife Service for excluding the Ozarks. But our subcommittee pointed out that, in fact, there had been no ecological and genetic surveys done in the Ozarks. All of them had previously been only in the Great Lakes region. And we thought that we should at least test these assumptions that they're having to exclude the Ozarks. We should do ecological and genetic surveys before this plan is implemented. Well, we did get a delay of the implementation of this plan from the Fish and Wildlife Service, and we also got permission to do our studies. And this was non-trivial because to sample an endangered species, you really have to go through a lot of bureaucracy in this country. But we got permission, we found out we could do it non-destructively, and so everybody was happy. And so the Fish and Wildlife Service says, fine, go ahead, do your studies, but we're not going to give you any money. So it was up to us to find our own sources of funds. So we tried to get funds wherever we could, and we patched enough together to do these studies. And like I say, without the volunteers, because the field work here, the mark recapture studies that we're going to be doing are very intense. They take a lot of people out on a fen and they have to spend a whole week there. So that's their vacation often. And then subsequent weekend visits for the next couple of months. But we did these mark recapture studies. I'm not going to go into details here. I can go into them, but they're very detailed and they actually yield a lot of information about things other than population size. So we show, for example, that in these fen complexes where you have several fens nearby, a single dragonfly in a single day can go to more than one fen. Okay, and they do. So they're very strong flyers. So we have a system of marking them, and everybody had to be trained. All the volunteers had to be trained both in marking but also in capturing the dragonflies in such a way that you wouldn't hurt them by netting them. But anyway, we were able to do quite a large number of fens, and we reanalyzed the data that had been previously done in the Great Lakes region, limiting it only to the ones that did a proper mark recapture study, so we could analyze all comparable studies with the same software. We use Program Mark to do this. And here's kind of a summary of the results. Okay, on the average Ozark fen had close to 500 individuals on it. Okay, and this certainly was smaller than the average that we had in the Great Lakes region, but it wasn't nearly as much as most people thought it was going to be. Because as you can see, the area of the fens in the Ozarks is 2 orders of magnitude smaller than that of the Great Lakes. So we're talking about very tiny fens compared to what they have in the Great Lakes region. However, we have a much larger density by an order of magnitude in the Ozarks than we do in the Great Lakes region. So from our mark recapture studies, we found that the Ozark fen density is nearly 5 times that of the Great Lakes region overall. So the Ozark fens are actually, in one sense, their optimal habitat, at least in terms of the density that can support them. So the Ozark population sizes, like I say, turned out to be much larger than the Fish and Wildlife Service had believed because of the data that came from the Great Lakes region. And therefore, it did have great potential for genetic diversity. So we then did a genetic survey to look at that. Okay, our genetic survey was unfortunately limited only to mitochondrial DNA because we weren't able to get enough funds. This was rather irritating because the Fish and Wildlife Service had put money into the Great Lakes region and had developed microsatellites, but the lab doing the microsatellites wouldn't waste their money looking at a peripheral population. So we never weren't able to do it. But we did look at all of our specimens that we gathered, and museum specimens, ones mostly from the 1920s. And also we used the data that was gathered previously by the Great Lakes people. And so this slide just shows kind of a pie chart of it. So we found many, many haplotypes in the Ozarks. And yeah, the size, the area of the pie diagram reflects the sample size. And so here in the Great Lakes region, as you can see in the southern Great Lakes, and these are mostly extinct populations here. This is the Ohio populations. But in the southern Great Lakes, we have a lot of genetic diversity. But in the northern Great Lakes, there was very little. And this was the same pattern found in the microsatellites. So that was why the Fish and Wildlife Service had split it into this recovery area and this recovery area. So the genetic studies here, you can see that we actually gathered more data, had a larger survey than the people that done the Great Lakes survey. And of course, we also included the museum specimens to get an idea of how things were changing over time. And this just shows the data we've got. And as you can see, we have very different sample sizes, and this creates certain statistical problems. So because that, I wrote a computer program that would subsample the larger populations so that they were the same, quote, sample size as the smaller populations. And we would do this at random, at least 1,000 times, sometimes more. And we would measure certain kind of classical measures of genetic diversity, such as expected heterozygosity. That is, what's the probability of two haplotypes being randomly drawn from your sample being the same or different? If they're different, that's, quote, heterozygosity. And also we just looked at the number of haplotypes as another measure of genetic diversity. So we used this program to correct for the very unequal sample sizes that we had in these three major populations. And we also divided the Great Lakes into its southern regions and the more northern regions, because as you saw before, they were very different. But it didn't really make much difference. So the conclusions we made was, we found when we compared the Ozark populations to the extinct populations that there was no loss of genetic variation at all. They all had comparable levels of genetic variation. So we had no evidence of loss of genetic variation in the Ozark population. However, the Ozarks did have significantly more genetic variation than any of the Great Lake regions, either considered north or south, or even as a whole. So if you combine all the Great Lakes regions together, this tiny area of 100 kilometers in the Ozarks has more genetic variation than the entire Great Lakes region. And we also found that the Great Lakes region, when you look just at the current populations, either just the southern region alone, there was a significant loss of genetic variation over both time, that is looking at the current Great Lakes versus the extinct Ohio populations, and space, as we saw as you went from the south to the north. The farther you got away from the Ozarks, the less genetic variation we found in Heinz emerald dragonfly. So that showed that the assumption of the Fish and Wildlife Service was completely wrong. In fact, it wasn't just that the assumption was wrong, it was actually reversed. It's the Great Lakes region that is genetically depauperate compared to the Ozarks. Another thing you can do with mitochondrial DNA, it's easy to construct what we call a haplotype tree. So we had an outgroup here, so we have temporal polarity. Each of these lines represents a single mutational change in the mitochondrial DNA. And a circle here represents an inferred haplotype in the tree that was not in the current samples. And then the current samples are shown in these larger colors and all labeled with a letter that corresponds to those letters that you saw before. So here was our haplotype tree. And one of the things we can do with a haplotype tree is we can do a nested clade phylogeographic analysis. So in this, we start at the tips, move one step in, and we form a union of all the haplotypes that can be joined in this manner. So all these are just, say, one step from A, so they form a clade or a branch. And you just keep doing this And then once you got these, what we call one-step clades, you just use the same algorithm. Now start with this as a tip clade and go inside. Here's another tip clade. So this becomes a two-step clade, and here's another two-step clade. The reason you do this is that this results in statistical independence of the contrast among the clades. So it's very easy to correct for multiple testing in this with this system. And you can also see the distribution here. Now, the nice thing about the nested clade analysis is, like I say, there's time here. For example, we know haplotype I is younger than haplotype A, or haplotype Q is younger than A, because time is flowing from the outgroup. And therefore, this is a younger haplotype, the P is younger than G, and so on. And similarly, we can see that this whole two-step clade here is closer to the root than this one. So this is the older, higher order clade, and this is a younger clade right up here. So we have temporal variation captured by this nested design, and we can make temporal contrasts. We now overlay on this the geography of what we know about the distribution. So we can measure, so for example, how widespread is haplotype Q? We can measure that. We can also measure how widespread haplotype Q is with respect to the entire clade in which it's in. How is it distributed relative to all the other members of its closest evolutionary relatives? And we can also look at temporal contrast. So we can examine, for example, how are these tip haplotypes distributed relative to the interior, the older one in this clade. So we can make many summary statistics that capture geographical information, but also use the temporal information that's inherent in the nested design. And then we test the significance of these by an exact permutation test. And when we look at the significant results of these summary statistics, they form patterns. And we have a way of making specific phylogeographic inference from these patterns. And our inference key has been evaluated both by computer simulations, which showed that in fact it has high power and is not prone to false positives for any of the kind of phylogeographic inferences made in this analysis. Moreover, we tested it against 150 actual data sets where there was outside information about a phylogeographic event or situation, and it did very well. So this is, I think, the only phylogeographic technique that has been validated both by computer simulations and by testing against actual data sets with known a priori effects in it. And it's done extremely well in both cases. So we could also do this nested clade analysis on the mitochondrial haplotype tree in the Heinz emerald dragonfly. And when we did that, we got several conclusions. First of all, I'll just talk about the Ozarks. We found that, in fact, there was isolation by distance, so that these fens up here in the upper region of the Black River, they were all genetically interconnected, but of course, the closer they were, the more gene flow there was. But there was gene flow interconnecting all of these haplotypes here, all of these populations. And I think that's one reason why the genetic variation was so high in these populations. They're not just one fen. Because there's so much movement of animals, you get many fens together, and the total population size here is much bigger than anything we see in the Great Lakes. We did see, however, a significant fragmentation event. This area here had been thoroughly checked, but there are no existing fens. The fens have been totally destroyed, mostly for agricultural purposes. And so there's a big gap here. And then there's just these other populations on the other side. So we had a significant fragmentation event that seems to have eliminated gene flow. So we do seem to need intermediate, a chain of fens to create movement of these dragonflies across space. And that was a very important conclusion that we had here from the Ozarks. But the really interesting conclusions are at really when you look at the higher level clades and include the Great Lakes. Interestingly, the fen complexes that we've had in our extinct population, even though the sample sizes were very small, the signal was still very strong for isolation by distance. So it really fit that pattern. So we knew in the past, these populations did experience gene flow. And remember, they had a lot of genetic diversity. We found no evidence of gene flow anywhere else in the Great Lakes region. And this isn't too surprising because, in fact, habitats here, the fen habitats, have been really thoroughly destroyed throughout much of this region for agricultural purposes. So all of these black dots are well separated by great distances with no fens in between them at all. So just like the Ozarks, and this shows, once you've eliminated those intermediate habitats, you destroy gene flow. And now in the Great Lakes, we have basically a bunch of isolated fen complexes that are no longer interconnected. So habitat loss is a major problem here in the Great Lakes region, and also in Missouri and the Ozarks. But the interesting conclusion is we had a significant evidence, a very highly significant evidence of a population range expansion from the Ozarks into the Great Lakes. So in the phylogeographic analysis, it indicated that the entire Great Lakes region, including the extinct populations, was actually a relatively recent founding event from the Ozarks. The Ozarks was the ancestral population, and it established this peripheral population in the Great Lakes region. And moreover, we did some statistics. We used a statistic called Tajima's D, indicating that there was really not a bottleneck associated with this. It was movements of large numbers of individuals going from the Ozarks into the Great Lakes region. And so there, you had plenty of genetic diversity. In fact, we saw the genetic diversity here in the extinct populations, and here was the same. Now, this is not surprising, or at least it shouldn't have been, okay, even to the Fish and Wildlife Service people, but it just never occurred to them. Where were the fens in the Great Lakes region 20,000 years ago? Okay, well, this shows, here's the Great Lakes region, and this shows where the ice, the glacier was. And in fact, 20,000 years ago, there were no fens in the Great Lakes region. In fact, there were no Great Lakes in the Great Lakes region. Everything was under a glacier, okay, at the end of the last ice age. And it was only with the melting of this glacier and its retreat that the Great Lakes formed and the fens formed. And moreover, as it did, there was much potential fen habitat in this region between the Ozarks and the Great Lakes. So knowing what we know about the current ability of these dragonflies, they could have easily dispersed in large numbers and colonized this region as fens formed in the Great Lakes region, okay? And this was relatively recent. Like I say, 20,000 years ago, there were no fens. And in fact, this work actually extends because it says the entire fen communities in North America were really derived from the Ozarks. It's not just this dragonfly, it's the whole community, okay? They came from this little tiny area right here, and now they're the ones all over the Great Lakes region. And this is also confirmed by the distribution. We said, for example, that this is the older clade, here is the younger clade, and there was a highly significant deficiency of haplotypes from the older clade in the Great Lakes region compared to the Ozarks, once again, when you correct for sample sizes. And so the Ozarks also has greater phylogenetic depth of its haplotype diversity than the Great Lakes region. The Great Lakes is dominated by a lot of very, very recent tip haplotypes, okay, much more so than the Ozarks, which has a much more thorough mixture of both old and newer haplotypes. Whoop. So in one sense, we totally reversed what was core and peripheral. We said, okay, for example, the Ozarks is really the core population ecologically, because in fact, they're the ones who are better adapted to their local fens. We also said the Ozarks are the core population genetically because they have significantly higher genetic diversity than the entire Great Lakes region together. We also said the Ozarks has the higher phylogenetic diversity. It's enriched for older haplotypes. And also, phylogeographically, it's the core because it's the ancestral population. In contrast, the Great Lakes populations are a recent peripheral population derived from the Ozarks no sooner than 13,000 to 8,500 years ago, which is when pollen cores and other things indicate the fens first appeared in the Great Lakes region. And therefore, our main conclusion here is geography alone, particularly in the current distribution, is not a reliable indicator of core peripheral status when climate changes. Now, of course, we all know we're in an era of climate change right now. So these so-called peripheral populations may be very, very important, even more important in the future than they have been in the past. But anyway, when climate changes, our current geographical distributions don't really tell us very much about where the important genetic diversity lies in these endangered populations. And this gets to the theme that I'd like to talk about that Martin mentioned about trying to overcome this idea, well, everybody knows. Okay, I find that in conservation biology, we're here to preserve biodiversity, but we often use general rules, the rules that we think are general, that ignore diversity. One rule fits all. And in fact, this whole idea that populations in the central core of the geographical range are the most important for species survival, that's something I've encountered many times in my long career in conservation biology. And the Heinz emerald dragonfly is just the most recent example. We really need to study the details of the species and communities we're concerned about. And here we're talking about not just Heinz, but the entire fen community, plants and animals in North America. Another very common thing is that hybridization is destructive to species integrity and should be prevented, and hybrid populations should not be protected. In fact, if you read the law, the Endangered Species Act in the United States, it actually prohibits the use of hybrid or admixed populations, particularly of vertebrates, okay, that you can't use them. They're not to be protected, they're not to be used. And here I'll just give one example of where we ignored that rule. I've ignored it many, many times.
Please, Sarah, mute your
Okay.
mic.
Okay. Anyway, I just recently got back from Israel, and where I'm working on the endangered wild ass, the Asiatic wild ass. This is not the progenitor of the donkey. That came from the African wild ass. This is a very different species, very different behaviors, ecology, and so on. Anyway, this species here, the Asiatic wild ass, went extinct in the Negev desert, and in fact, throughout the Arabian Peninsula, many other places in the Middle East in the 1920s. After the collapse of the Ottoman Empire, a lot of people moved into areas in these desert areas, and where do they go? They go where there's water. And the wild ass, the females, when they have their foals, they need to drink water fairly regularly. And there's only a handful of places in the Negev in these desert regions where they could, and now they were being excluded. Moreover, people found the flesh of these tasty. Okay, I can't swear to that myself, but anyway, they were hunted. And so by the 1920s, the Asiatic wild ass was pretty much extinct in this entire area. There were a few populations in zoos, particularly in Iran. And an Israeli investigator was able to get several animals, about a dozen animals, out of Iran before the fall of the Shah. And they brought them to Israel, where they started breeding them. However, people objected to this because the zoo and Iran had actually mixed two different subspecies of the Asiatic wild ass, and the only animals in existence now were hybrids between them. And some people said, wait, you can't use hybrids, these are useless. So they were having this strange argument that it's better not to use these hybrids and let the animals be extinct in the wild rather than make use of the hybrids. But we ignored that. So starting in the 80s, we started releasing these animals into the Negev after habitat restoration and ensuring that there were water holes that they could access to that were not, that were in remote areas, that would not be, where they wouldn't be hunted or affected by the exclusion. And I'm glad we did it. We're now doing a lot of studies on these animals. We've got the full genome now sequenced, and we can reconstruct pedigrees in the wild population. It's going to be a tremendous resource for conservation biology. And the animals are doing extremely well. Moreover, the Israeli program attracted attention of people in Saudi Arabia, and now they have their wild ass program that's also based on the Israeli program. And this had to be done in secret at first because of politics, but now it's actually openly acknowledged in Saudi Arabia about the help that came from Israel. So the population is doing extremely well, but we wouldn't have done this if we had listened to the thing that, quote, everybody knows, hybrids are bad. And in fact, modern genomics has shown that hybridization and admixture are occurring in all over the place, including humans, okay? Our own genetic ancestry has been profoundly affected by admixture. And this is true for many other species. So this whole recommendation is so false now, it should be totally abandoned. But there's still people, it's an easy rule, and people still use it. Another one is that a species can't be saved unless it satisfies the 5,500 rule. The 50 says you have to have at least 50 animals to start the population, and you have to grow it to 500, at least 500, to preserve genetic diversity in the long term. And if you can't satisfy this 5,500 rule, it's basically a hopeless case. And this was actually the first conservation project I got involved in the 1970s with the Spix gazelle. This is a little gazelle that lives, has the most geographically restricted range of all the gazelles. It lives only in a part of Ethiopia and Somalia. And in fact, the latest reports indicate all the Ethiopian populations are extinct now. And the Somalian populations are hard to study because of warfare that's been occurring for 60 years in that region. But anyway, it's a highly endangered species as far as we can tell, even now. And it does have this inflatable nose that it uses as a warning sign. But anyway, they're beautiful animals. And back in the 60s and early 70s, the director of the St. Louis Zoo brought together all the animals that were in the zoo records all over in North America and Europe and collected all the animals to the St. Louis Zoo. And all the animals that were available that he could find were 3 females and 1 male. So obviously this, to most people, did not satisfy the 5,500 rule that you have to start with at least 50 animals. And in fact, a new director came as the old director retired. And the new director said, we can't waste resources on this population, this captive population. It's because it has no hope of winning. It has no hope of survival. It violates the 5,500 rule. And so he decreed that they would stop breeding and just let the animals naturally die out. Okay. Well, fortunately, the curator of large animals didn't believe this was true, and he contacted me. And so we designed a program, and in fact, it was very successful. And we've been able to breed these animals, cooperate with other zoos throughout North America and Europe. And now there's a very healthy population of these animals. Fortunately, we ignored the 5,500 rule. And in fact, the 5,500 rule, I think, has caused more damage than not. Okay, I've had people use it as an excuse to let populations go extinct on more than this occasion. And moreover, the 50 and the 500 are not actually the number of animals or individuals. These are these strange things in population genetics called effective population sizes. And in particular, the 50 is a inbreeding effective size, and the 500 is a variance effective size. And these can have very little relationship to the actual census size. The 50, by the way, was based on the original author who proposed this rule on his informal talks with animal and plant breeders about how much inbreeding they could take. And he said, oh, it's 50. Not a supporting reference at all. The 500 comes from a single experiment in Drosophila melanogaster, one strain of Drosophila melanogaster on the trait of sternoplural bristle number. That's it. That is the entire data that was used to justify the 5,500 rule. And biodiversity all over this planet has been affected by this very limited database. And like I say, effective sizes can be very different from what you census size. So Stan Browdy and I actually went to the trouble of estimating inbreeding and effective and variance effective sizes for different rhinoceros populations, another species of great interest. And as you can see, I'm not going to go through the details, you can see that in fact, there's very little relationship between the census size and the inbreeding effective or variance effective size. Moreover, another general rule that everybody knows is that effective sizes are smaller than census sizes. Well, here you can see that is certainly not true here for the black rhinoceros, okay? Now, it is true here for the southern white rhinoceros. Sometimes it's true, sometimes it's not, okay? And here we have for the northern white rhino, which is very highly endangered, notice that even though there's only 23 animals, the inbreeding effective size does satisfy the 5,500 rule. But of course, they're nowhere close to satisfying this. They're just the beginning of the breeding program here. So to tell the truth, I have not seen a single actual implementation of the 5,500 rule where people actually went to the problems of solving and estimating inbreeding and variance effective sizes rather than using census sizes and shaping their conservation program on that basis. So this is another one of these rules that everybody knows that I think we would be better off just abandoning and dealing with the specifics of the endangered species that we have at hand. And so with that, I'll stop and just wait for some questions if you have any. I'm happy to answer them.
Hi, Alan. Thank you so much. This is a wonderful work, and it was a wonderful talk.